And the next speaker is Niels Averes. I hope I pronounced the name well. Niels is postdoc at Stanford University and a part of NASA's Space Technology Research Initiative, CUBES, where he is developing microbial cell factories to produce materials and consumables. He is also an analog astronaut. He recently came back from a Mars habitat in Hawaii, where he was testing biomanufacturing capabilities in the field. Great to see you, Niels. I hope you will have a chance to tell us a little bit about the HI-SEAS too during the meeting.
Thanks. Yeah, thanks for the introduction. I'm glad to be here. Yeah, let me try and share my slides. Where did that go? You can see my screen.
Yes.
I'll go with that. That's the best we can do right now.
Perfect. Thank you, everyone, for waiting. And please go ahead.
Yes, really sorry about the delay. Great to be here, and I hope this works now. And I'm glad to speak on synthetic biology for space exploration, specifically also about my microbial cell factory for production of high-performance materials. What is space synthetic biology? So this is my attempt at a definition. Space synthetic biology, the development of biological technologies that can increase the capabilities of humans during exploration of deep space. And I am part of CUBES, which is the Center for Utilization of Biological Engineering in Space, where we do exactly that. And CUBES is a NASA Space Technology Research Institute, and its objective is to support biomanufacturing for deep space exploration that realizes the inherent mass, power, and volume advantages of space technology over traditional abiotic approaches. And that is all in front of the backdrop of sending eventually a human mission to Mars. When going to Mars, the mission scenario is much different. So while to the moon, which is relatively easy, you can go there within a couple of days and come back. When going to Mars, you have to go there and stay for over a year until the orbits align again, and you can hop back basically to Earth. And that basically changes everything, because currently we're bringing everything on missions like that. Even to low Earth orbit, we're bringing everything that the astronauts need. And for such a long time, you obviously have to bring much more. And also because it's so far away, it's much more expensive to bring everything. While the launch costs to low Earth orbit are about in the tens of thousands of dollars per kilogram, that is 10 times more expensive when sending something to Mars. So the real limiting factor here of exploring Mars with humans is the cost of launching materials into space. Because, yeah, it's inherently dependent on resupply. And one potential solution to that could be in situ research utilization. So basically using what you can find where you go to produce supplies like food, fuel, oxygen, and materials. And for that purpose, CUBES is deploying biotechnology as a means for in-space manufacturing. So what can we find on Mars? The regulus there, the soil that is, contains elements that are vital for living organisms. And the surface is quite oxidized, and has no fixed carbon so far been found. No crude oil, very most likely. So all the industry that we have here on Earth, you can't really feasibly think of that on Mars. The density of the Martian atmosphere is pretty low, 0.6% of the Earth, so 6 millibars, but it is composed mostly of carbon dioxide and some nitrogen, which equals 25 trillion metric tons of CO2. So I have carbon that we can work with. Also, there's water. There's quite a fair amount of water, actually. Water is present on the polar ice caps with a mixture of 85% carbon dioxide ice and 50% water ice. And basically, it has been estimated that in total there's enough lake to fill about half the Baltic Sea. Total of 5 million metric tons of water. So we have water and carbon dioxide and sunlight. So basically, we could potentially grow plants. However, perchlorates are also widespread on the surface. And these are toxic compounds which are potentially a source of fixed oxygen, but they're problematic for living organisms. They compete with nitrates in plants and thyroid iodate uptake in humans. So would potatoes grow on Mars like in the film The Martian? There have been experiments with crops growing them in equivalent amounts of perchlorates on Earth, and these plants grow, but the perchlorates enrich in the leaves mostly. So the question would rather probably be, could Watney have survived on plants from Martian soil? However, crops are a suboptimal use of resources anyway. They have a low biomass density per footprint, per volume, and a very slow growth rate. They're pretty demanding in terms of their cultivation conditions, if you think about all the nutrients they need, and they're not very tractable. A plant is basically a plant. So the solution, or the idea, is to turn to microbiology. Like that, for example, growing cyanobacteria in huge ponds to produce an initial biomass from the carbon dioxide and the water that is present on Mars. But what should we produce? What do we produce? I think very important are materials, because basically everything you build here has to be somehow contained. The domes, the ponds, even the astronauts themselves, their spacesuits. And that is what I'm looking at. The materials used to build spacesuits or spacecrafts. These are often polymers, like, for example, Mylar, Dacron, Kevlar, Vectran, polyesters and polyaramides. These materials need to be really high strength, ideally also low weight, and thermally as well as chemically very resistant. Certain aromatic polyesters fulfill these requirements. Like I said, Kevlar and Vectran especially. And that's what their structure looks like. They're aromatic polyesters, and they're also used on Earth, for example, for sailcloth or insulative foil like Mylar. It's basically what you find in these first aid kits, these thermal blankets. And these materials are really strong. So the idea is, or the question is, can we produce these biologically? Because currently they're still produced from crude oil here on Earth. And that brings me to the next part of my presentation, where I go deeper into the biomanufacturing of the high-performance bioplastics from CO2. So what we can make are polyhydroxyalkanoates, which are natural polyesters. And they are produced by a range of microorganisms naturally, and they can be obtained in a one-step bioprocess already from carbon sources like CO2. They're 3D printable, moldable, and thermoplastic biopolyesters, but they're not quite that strong. They have a thermal decomposition temperature, which is really close to their melting point. It makes them hard to process. So if you melt them, they quickly also break down, which is not ideal. Especially the brittleness is a problem for their processing and for their application. What about if we could make polymers like that, where the aromatic ring is in the backbone? That's basically what makes these materials that I showed earlier strong. The aromatic ring, the bulky group in the middle of that polymer backbone. Comparing the properties here, for example, of PET and Vectran to PHB or even PLA, PET and especially Vectran, they're much superior. So can we get to these materials? And how? Apologies for this slide. It should have been a nice animation. So basically, I'm not sure how recognizable that is. Let's go with this one. PHAs or polyhydroxyalkanoates, biological polyesters with aromatic rings in the side chain exist. They're based on monomers like phenylactic acid and mandelic acid here on the right. And yeah, the closer the aromatic ring, the stronger the polymer. But how do we get that aromatic ring into the backbone? I have found one compound that could work. This is fluoridic acid here on the bottom. And using these three monomers, we could get polymers that look like that. Like I said, the first two on the right top, they have already been shown to be bioproducible in an engineered system with Escherichia coli. So using sugars to produce these materials. And what I'm looking to do is implementing this pathway into a gas fermenting organism that can use carbon dioxide as a feedstock, while also extending it so that we can get these polyarylate, these materials with the aromatic ring in the backbone. And for that purpose, I deployed a metabolic engineering strategy that looks like that. So the natural pathway to production of polyhydroxybutyrate looks like that, and I'm entering it on the side. Basically, I added to that genetic pathway a new PHA synthase. That is the polymerase that extends the the polymer chain, as well as a CoA transferase that initially loads the monomers that I just showed on CoA, so that they're available to the PHA synthase, the polymerase. So the engineering strategy basically involves entering this pathway on the side by swapping out the PHA synthase with a more promiscuous one that allows us to polymerize these aromatic monomers, and then adding also the CoA transferase. So I engineered, those are the enzymes, the HAT-A and the Fas-C. So I engineered Cuprividis necator, a gas fermenting organism, and introduced those two genes, that pathway, into that organism. And basically I tested what we could get. I supplied a bunch of precursors, different aromatics as well as aliphatics, to see what the polymers are that we can get from that organism. And we had success, a lot of success with phenolic acid. Initially we tried without any precursors, and we could see that the introduced enzymes could rescue a PHA negative phenotype. So we had knocked the natural PHA synthase out and supplied the heterologous one, and just introducing it allowed the strain to already produce PHB. The yield was similar to the wild type, which is great. So we're not there's no drawback there. And the polymer is mostly constituted of poly-3-hydroxybutyrate. That's the natural polyester, but with some minor fractions of other compounds. So we're getting something different there. Then we tried feeding compounds like phenolactic acid, which I said we were successful with, and we're getting a pretty high incorporation ratio here, which is great. So we're getting about 33% of phenolactic acid incorporated, which alters the polymer properties. It's not brittle anymore at all. Like when I extract that polymer and just extrude it, dry it in the lab, it is pretty soft. So that already changed and optimized the polymer properties. Other precursors like mandelic acid were only obtained at a lower fraction, but still we could incorporate those. And then I tried the really novel thing, trying to get fluoridic acid in there. And what we got was, surprisingly, this very yellow polymer, which is not known at all. Like PHAs are mostly colorless, but we got this yellow compound, yellow resin, which also turned to be hardly soluble in any of the common solvents. So that already indicates a change of polymer properties and higher chemical resistance. And through GCMS, we were able to confirm presence of fluoridic acid in the material. So that's a success. We somehow got that compound in there. So NMR, we were also able to then ultimately validate that there is fluoridic acid in the backbone of the polymer. And we're able to determine the content, which is 1%. Not much, but like I said, it already alters the polymer properties. And yeah, this is the first step, the first push into this direction. So not sure what the next slide is. Yeah, those are the polymers that we can currently obtain from our engineered microbial cell factory. And the next step would probably be to increase the content of fluoric acid in that polymer and optimize the properties. But what I just did was try and see if we can at all use this microbial cell factory for remote biomanufacturing. So I did some field testing with those engineered strains on an analog mission at HI-SEAS, which is HI-SEAS is a Martian simulation on the slopes of the volcano Mauna Loa. So it's a space analogous habitat. So it's supposed to simulate what a base on Mars could look like. So the mission that I went on took place in August this year, and basically was a 2-week mission where I demonstrated and prototyped the technology to biomanufacture plastics in a remote location. And we were constrained pretty much by everything: water, power, the conditions that I could use in the lab. Like I couldn't run sterile conditions, for example. So I was pretty restricted in the resource conditions as well. No biosafety cabinets or fancy laboratory equipment there. And that is what HI-SEAS looks like from the inside, the dome. It is fairly spacious, I would say. Still, it gets, after a couple of days, seeing only the same one wall, basically, because it's a dome, it gets tiring. Yeah, there's an upstairs area where everybody has their own bedroom, and downstairs the office area, which also serves at the same time as basically main living area as well as exercise area, and the kitchen is just around the corner. Yeah, sometimes you are sitting there on your desk and working on your reports, while others behind you are working out, exercising, and doing their daily routine. So it's really great if you can get out, which is called extravehicular activity in an analogy to a real Martian mission. So we put on these mock-up spacesuits and go through an airlock cycling process to perform ground operations outside. Here's me outside with another crew member using ground penetrating radiology to investigate the lava tubes there. And yeah, but it's not always that nice outside, so you're lucky when you catch a day that is has these blue skies. Often you're just sitting there and looking through your one single window, which is by the kitchen, and watching the dust storms, which is basically rain. So all you're left with is turning to lab work, and that is the lab at HI-SEAS, which is, like I said, a basic laboratory with pipettes, a stirrer plate, but no large appliances like incubators, fume hood, or a laminar flow cabinet. Nevertheless, I tried to make it work, and I brought some basic bioreactors where I tested my microbial cell factory in a remote biomanufacturing experiment. I had basic temperature control, aeration, but no regulation of aeration, no regulation of pH, only qualitative monitoring of growth. And objective was basically to see if under these circumstances we could still get those bioplastics. And yeah, the answer is it does work. So we can operate these bioreactors there under semi-sterile conditions, and microbes grow over pretty much the same number of days. That's as specific as I can get for that kind of experiment, as it gets for this kind of experiment. And I was able to extract the bioplastics here, and yeah, so that can be called a success. You can really use this technology in remote conditions. But in the end, it's always still the best if you get out with your crew members and explore the surrounding lava tubes, which is just an awesome landscape. There are these, it's all lava, and if you manage to get to the lava tubes, you're rewarded with this pretty much private cave that you can explore. And there's interesting microbes going there as well. Other researchers are working on that. With that, I'd like to end my presentation, and I'm certainly open for questions if there's time.
Super, Niels. That is very exciting. We have one question in Q&A chat. I'm going to read it to you. Maybe you can see it there too. So Leo Penny is asking, catalysts are often used in creating specific organic materials. Has there been any investigation into using reusable catalyst plus biological pre-post processing, sort of a hybrid between chemistry and biology?
I'm not sure. I don't work on the chemistry side.
Mm-hmm.
I don't have a good answer for that, yeah.
Okay. You said that the atmosphere in Mars is only 1.6% of the Earth atmosphere, and there is a lot of carbon dioxide, but still it seems to be pretty diluted. So if you want to grow organisms, would you have to concentrate carbon dioxide? Would it be just, what's the solution?
0.6%. So yeah, you have to compress the atmosphere, of course, because at 0.6%, that is below the Armstrong limit, which means that there's no liquid water at 0.6%. So yeah, you'll have to compress that atmosphere. And there's a whole chain of technical solutions for how to convert that CO2 into oxygen as well for the astronauts to breathe. Yeah, first step is of course compression, and there are these technical solutions which are of course, or which are superior to any biological solution that you could take. There are approaches to directly use that atmosphere without pre-processing, just directly compress it and basically feed the pure CO2 to cyanobacteria. So that has been looked at. But yeah, I would say there's niches for technical solutions and there are niches for the biological solutions. And the real thing, the real paradigm would be to combine those two in the most beneficial way. So yeah, compress that atmosphere, process it to closer to Earth, and then you can use that for different purposes, for microbiology, biotechnology.
Thank you, Niels. That was a really interesting presentation, and thank you for telling us also about this analog mission. It really it seems pretty amazing what you did. So thanks a lot, and we will move now to another speaker, that is Kate Adamala. Thanks.